So, hi. So I will try to compress in a few slides what was a very complicated work. What was complicated because the big question we were trying to answer is contribute to answer is how currents disperse plankton, and we did this in a sort of a multidisciplinary approach with the molecular biologists, physical oceanographers, and ecologists and numerical modeler. And we focused on a specific point, a critical point, most critical point of the global circulation. The work was fully multidisciplinary, maybe a premiere in literature, but I want to emphasize this for many reasons. One is that probably it will be too long in the presentation. The second one is that any one of my collaborators, so we worked really together, Émilie Villard, who was the postdoc in charge, the first name, and Fabrice Not, Pascal Incamp, and myself. So any one of us could have done this presentation, but maybe with a different flavor. This is what is special of this situation. It was very hard to work together. Sometimes it was beautiful. Sometimes we are one physical oceanographer, one biologist, one bioinformatician, and a specialist in eucalyptus, actually. So the situation, sometimes we exchanged role, which was great. Sometimes the situation was so messy. It was like, the old joke on hell. Hell is made of place where police is German, cooking is English, lovers are Swiss, and everything is organized by Italians. So that was a kind of the situation somehow. So first a few short words of introduction. So the main point here was we have the oceans organized into, this is chlorophyll, where regions where are very low chlorophyll, which are called subtropical gyres, okay? And there are regions where you have on average much more chlorophyll. And this is because basically here, nutrients which in the ocean are deep, they do not reach the surface, so where is light. So there is a little photosynthesis there going on there. And this is opposite to regions, subpolar regions, where you actually nutrients can get to the surface at least sometimes over the year and create a different environment. So our question was how those big gyres are connected among them, if they are. And we have to also keep in mind the global surface circulation. So you see the gyres all over, but you also see that generally at surface the circulation goes from east to west. Okay, this is an important point. And then water sinks here, goes deep, and emerges almost everywhere. So we have a direction of the flow, and so our question was, and as you can see, the choke point of the surface circulation is actually here. So all the water coming from Indo-Pacific Ocean that goes to Atlantic has to go through here. So it's really the big choke point of circulation. So we focused on the question, how, as this is a place where everything passes through, or there is any kind of filtering there of the diversity. And we worked at an end-to-end scale, and focused on this region. So, more broadly speaking, we attack this question because the ecosystem, the ecology, and the evolution of a system is very different if the system is isolated or it's within a community, within a system of community, and it's broadly connected. So, from an ecological point of view, basically, the point is that if you want to have a response to climate change, the resilience of the system will depend on the diversity of the system, so how many possibilities of response you have, basically, on the adaptive capacity that can be acclimation, can be evolvability, but also the connectivity. So if you are in difficulty, one species is particularly vulnerable, from another system you can have an invasion of another species which is more resistant. That will allow you to resist. From an evolutionary point of view, of course, for large populations, the gene flow actually between populations is the main term in general for evolution. So basically the question is, are the oceans a system of isolated ecosystem, or there is a pan mix here, or we are in between the two? But the challenge is extraordinary because actually in the ocean everything is moving all the time on different time and spatial scale. Several biological processes involved, which we don't know basically. Schematizing very quickly, so the point was we have an ecosystem here, we have one element which is vulnerable to climate change. For instance, if we have another ecosystem close where one of the similar species is actually less vulnerable to climate change, this species, if can pass, can replace this one, okay? So it's more stable. Of course, how to jump from one to another one is another big question. And what we focused on is basically on this point. Is the passage between the two ecosystems actually a true passage? And second point is this place vulnerable to climate change? Is something we have to take into account to understand climate change response? So for doing this, first of all, we plan sample in the area from the beginning, before the expedition, in a way that we have a good description of the source region in the Indian Ocean and in the Atlantic Ocean. So, and the result of a comparison with very many methods of the diversity in the different samples is basically that the similarity in terms of richness between the two basins is about 30–40%, okay, between the two basins, while the Southern Ocean, as previously hypothesized, is actually much more isolated because the similarity between the Southern Ocean and the rest is less than 10%. Then we tried to, the one problem with this study was that there are basically very few studies of this kind for the ocean. So we were somehow forced to derive our own conceptual framework. So drafting from the actual debate in coral reef diversity, we derived this kind of general schematics, where here, what you see here, it's how we distribute the different OTUs between the two basins in this kind of schematics, where here on OTU or group is a picture where the x-axis you have the Indian OTUs found in the Indian, which were specific to the Indian, divided by all the ones found in the Indian Ocean. So if all the one found in the Indian Ocean were unique to the Indian Ocean, this will give you 1 here. But if the one found in the Indian Ocean were also in the Atlantic, they were not specific to the Indian, so they were 0, okay? The same here it's for the Atlantic. The interesting point here is that with these schematics, you will have that a group that appears here means that it's not specific to any basin. So this is something that is coherent with the everything is everywhere, okay? Or, in other words, that there is a perfect mixing between the two populations, a pan-mix here. Here, if you have a point here, a group is mostly here, means that there's many OTUs which are specific to the Atlantic, but very few which are specific to the Indian. So that means that in the Atlantic accumulates a local source of richness plus what is transported from the Indian, okay? Those are two cases are also very interesting. Here, a point is made here, a group here is made of OTUs which are both specific to the Indian or specific, or the Indian specific to the Atlantic. That means the two populations are different, are well separated. And if you have something here, that means that it's mostly made of OTUs specific to the Indian, while the Atlantic OTUs are similar to the one of the Indian. So that means there was a barrier here. So even if there's a physical transport, there is a sort of barrier between the two. Using these conceptual schemes, I'm summarizing the work. I mean, we have many details, but summarizing overall, this is the results, which is kind of surprising for us. So basically, only 4% of the OTUs in this comparison of reads using 18S is actually appear to be everywhere. Only 19% gives the idea that the Atlantic is a basin of accumulation, so which corresponds to the idea that we have a diversity, species popping up all over the oceans and then accumulating because of the circulation. And most of the cases actually goes into the case in which the two structures are quite separated, or that the Indian is actually richer, which corresponds, which nicely fit with the idea that Indian and Pacific are wider, so you have most species, stochastically speaking, popping up. But then there is a sort of filtering when this is transported to the Atlantic. So there is something going on in the Agulhas region, something going on in the region between the two basins, and so we actually sample what was the main process that causes the transport. This is also because there is a deep oceanographic interest in these features, because if you look in detail, here's Madagascar, is Africa, so the current goes like this, but actually the head of the retroflexion detaches and forms the huge rings called, which are huge structures, which are beautiful because they can live up to 3 years and it goes up to Brazil. They are very large and very deep. One layer, this is the ring compared to France, and if you actually pour the water of one ring over France, you will cover the entire country of 100 up to 500 meters of water. So they're huge stuff. They transport heat and salt and stabilize the global climate. And during glaciation, they probably were not formed. So during glaciation, those two systems were separated. Finally, it's important to say that the studies that demonstrate that there was no connection are based on the assumption that what you find in the Indian species found in the sediments here are actually transported by the rings. So in the old paleo studies on glaciation, there is the assumption the rings perfectly transport Indian species. Okay. Now, so what we found, we sampled two rings. We identified them by satellite before Tara went there. And what we did, basically, we sampled the rings and also the source water in the Indian. So the ring, one ring was here, and another one was close to Brazil. We described the community with several methods, with putting together a large group of taxonomists and experts. And also we traced the origin of the rings using satellite images. So we basically know that one is 9 months old, another 3 years old. And then we've had a nice discovery, which is the chemistry of the ring was very special. If you look at here, the profile, a vertical profile of nitrate, so NO2, you see that within the ring there is a very strange peak. So this is a transient tracer, okay? It's not the normal standard form nitrate you find in the ocean. So something was strange there in terms of biochemistry on the ring. But, one measurement, one time is not enough to conclude anything. So we interacted with Mick Follows at MIT, and we did a systematic analysis of how the rings are described in this global model of the oceans. So we were able to actually derive a sort of scenario that I will illustrate in a minute. And then, finally, we get back to genomics of the species to try to have a confirmation of this biochemical scenario. This was the source of the story of the study, and the results are basically that when we went into the ring, we found the water there was actually 5 degrees less than the original water in the Indian Ocean, so there were no more subtropical water in some sense. It was intense cooling. But above all, we found that the mixed layer, so the mixing in the ring, the vertical mixing, was now very deep. That is, the particles or the cells were moved up and down over scales of a few hours after 2 days on a scale that is comparable to the Tour Eiffel. And you have to remind that light in the ocean is only in the after first 100 meters. So that means the things were going into dark continuously. So there was a strong light limitation, but also nutrients were fished out from that. And so basically what happens was this one. So is that there is a strong storms going over there in the roaring 40s that cause a deep convective mixing in the rings. And then this causes a light limitation and nutrient entrainment for the deep layers, which cause a bloom of phytoplankton. But then there is because of storming, because of the intermittent character of the forcing, there are sequences of deep convective mixing in which now the phytoplankton is now pushed into the dark, okay? And there is remineralized, and this explains why we have this deep excess of NO2. All this, coupled with the cooling, cause an important community shift. So moving from the Indian to the Atlantic is actually a shock for most of the species. Just to illustrate a bit in a picture, more picture way, the sense of what we have found. So here is a simulation of the winter in the region. Okay, so you have Africa. You see this is temperature. This is primary production. So carbon, how much carbon is fixed, as simulated by the model. So you see the current, very warm current coming here. You see the formation of the agulous rings traveling here. And if you look at the winter, you see there is a lot of primary production going on here, because actually there is a mixed layer is taking nutrients, but you see the rings are blue spots, actually. So light limitation is there, is not allowing for response. And if you go to spring, you actually see that while in spring now, the subpolar region I described at the beginning is now blooming, as happens also here in North Atlantic. Now the rings also blooms too, okay? So this prompted us to this idea that actually rings are sort of subpolar environment traveling across the subtropics because of the particular physics. Now, just a couple of more words on the community shift was really important. Here you can see, for instance, how the ecotypes of Prococus change when moving from the Indian Ocean into the ring, where you have we see the appearance of a low light adapted ecotype of Prococus, which confirms somehow this light limitation, and in general, the dramatic shift from the Indian to the ring, which in principle is Indian water, because of these specific characteristics. But also, we came back also to validate this scenario where we couple physics and biochemistry. To validate this, we look into the anomalies of the presence of genes related to nitrate cycling in the ocean within the ring with respect to the source region. So what you see here is a schematics of what's the cycling of the nitrogen in the ocean. And what you see in red here are genes that were more abundant in the ring with respect to the original source water, and in green, which were less abundant. So somehow we demonstrate that there was a shift, a significant shift in the biochemistry of the system, because of the bacteria responsible for that were actually changing. So to conclude, we characterize the phenomenal geodynamics of the Indo-Atlantic connection, both at large scale and locally, and also the dynamics of the connection. And we found, basically, using this sort of new conceptual scheme, that actually are quite similar, but in fact, if you look at the fine scale, they are not similar completely, and there are an interesting difference that probably this is caused by the specific physics and dynamics of the Agulhas rings, which behave like a subpolar-like environment. And more in general, the point is that somehow we demonstrate that also for the oceans, we have to take into account the existence of metabolic barriers between systems. And I want to remind that metabolic barriers are very sensitive to climate change. You can have a change in storminess, so a change in the mixing, and so the passage could be impacted. To conclude, finally, the perspective, because we are intensively working on this data yet, there are two interesting points. One is that now we will have soon available the meta-transcriptome data about this same system, so we can look into the metabolic response of eukaryotes, and also that we can try to work at a more finer scale on genes to establish a biogeography of genes. And finally, the invitation, let's go back to Agulhas. Thank you very much.